Our topic is simple mislabeling correction. So, what is simple mislabeling? Is that the labels are misplaced by machine or human error. If this happens in a hospital lab, what it may cause? Let's see.
Mr. McKenna, you're cancer-free. The biopsy looked like adenocarcinoma, but it wasn't. Harmless lesions on your lungs. You're fine.
I don't get it. I just accepted an offer on my house. I've had 3 goodbye parties. I'm buying plane tickets to Venice.
You can still use those if you're alive.
I have to pay a $6,000 broker commission on a house I'm not selling. Money I don't have. Thank you for letting me know.
A patient was diagnosed with cancer, but it was not until 3 months later that the doctor told him that he was cancer-free, just because of disease mislabeling. The result is that the doctor was sued by the patient. Actually, this also happens in our lives. Mislabeling will make lots of troubles. So, this explains what the problem is of our project right now. We intend to make better labels classification. During the whole project, we used Apex Spark, a distributed processing system, to process all the data and explore our machine learning algorithms in Amazon Elastic MapReduce. First, we need to know that there are relations between RNAs and proteins. In the first step, from the left picture, the information in DNA is transferred to a messenger RNA, mRNA molecule by way of a process called transcription. During translation, which is the second major step in gene expression, the mRNA is read according to the genetic code. From this short video, we can see that proteins are synthesized through RNAs. The words GGU and the CCA can be seen as a key pair. So, if we find the relations from these two data, we are able to judge those mislabeling. So, first of all, we need to do some pre-process for our data for protein data sets. Each test sample contains more than 4,000 data points, and for RNA data sets, there are even more than 17,000 data points per sample. If we use all of these data points, the calculation is extremely large, even for Amazon Web Service. So, we need dimension reduction technology, like this sentence, to find out what Mike is doing. We don't need to read and understand every word in the sentence. All we need is the important words. Mike is reading a book. Dimension reduction can do the same thing to our data set. We labeled the data by male, female, MSI low, and MSI high as given in the clinic file. We pick the data points that can split the data by label calories to keep. After reduction, the size for both protein and RNA data size are minimized. We have RNA and protein data size from same 80 test sample, and their mislabeling happens in both data size. Now, we want to generate our own training data. What we do is take one sample data from RNA data site and combine it with all sample data in protein to form 80 new training data. And we label each new sample 1 if and only if RNA and protein are from the same test sample. All other new training data are labeled as 0. We do the same process to all sample in RNA data sites, which give us 6,400 new training data. 80 of them are labeled as 1 and the rest of them are labeled as 0. So right now we have the positive and the negative training sample pairs. We subtract 50 positive and 50 negative sample pairs to train this neural network. The output of the neural network means the matching probability of the given RNA series and the protein series. After training this network, we will get a model to help us quantify the matching degree. To avoid overfitting too much, we also applied drop-out method and got a result of accuracy over 95%. Although the outcome seems pretty good, we still have an issue about whether our trained model can be used on the test site. Because, as we know, the neural network works only the training set and the testing set have the same distribution, which is unknown here. Due to the fact that we only have 80 samples and generated 6,400 sample pairs in this project. Another way to do this matching problem is using traditional machine learning method, support vector machine with kernels. Support vector machine usually works better when training data set is not that big, and it also deals with the problem that the number of positive and negative sample pairs are not balanced. And this is the process of our final project. First, we do the analysis, then reduce the dimension, and then we find the relation between the data, and finally we got the result. And this is the related reference. That's all. Thank you for viewing.